Welcome to Red Light, Green Light, the podcast where seasoned leaders and top-tier executives in science, tech, pharma, finance, and innovation open up and get real with me and with you. You'll hear personal stories from key moments of their career arcs and how they've struggled and succeeded when it comes to moving audiences from the red light of risk to the green light of go. I'm Cindy Skalicky, storytelling strategist and author of the best-selling book, Red Light, Green Light, How Top Leaders Present with Polish, Get Buy-in, and Become More Influential. On this podcast, you'll hear from an exceptional invite-only lineup of leaders who've been in the room and at the table when stakes were high, decisions were hard, and their stories had to land. This is where expert minds share stories that shape the future. What's more, you'll walk away with tips you can try immediately to drive the vision of innovation with your team. Hi, everybody. Welcome. Welcome to another episode of Red Light, Green Light. I am your host, Cindy Skalicky, and every week I bring to you all a leader in science, technology, or innovation, in this case, pharma, who can share with us some of the storylines that person has experienced, learning the ropes of storytelling with deep rich topics like science, tech, and innovation. And today I have a wonderful guest I'm very much looking forward to diving into, Dr. Nidhi Arora. Dr. Arora, thank you for coming and spending some time with me today. Welcome.
Thank you so much, Cindy. It's a pleasure to be with you here.
Yes, you're so
Happy to
welcome.
be here.
Yes. Let me read for our audience like the quick little intro that I brought, and then we'll dive right into the red lights and green lights of what you do. Okay. Nidhi is a computational chemistry executive, right? I mean, are we all, striving for that, which is just fantastic. She's also a drug discovery leader with more than 25 years experience across organizations that will resonate and you will know Roche, Johnson and Johnson, Takeda Pharmaceuticals, and that's where she most recently led computational structural and protein sciences. So, Nidhi, when we talk about scientific communications and them being really complex, you take the cake with like, what is computational structure? What are protein scientists? That to me sounds like triple clicking on something. So help us understand what level of the science you love and why.
Love all of the drug discovery, but I trained in my PhD from Indian Institute of Technology, Delhi, in
Okay.
computational chemistry, and then came here to US Scripps Research Institute for doing a postdoc in
Okay.
computational chemistry. Since
Okay.
then, I joined Roche Pharmaceuticals as a
Mhm.
computational chemist, and then J&J, and as you just said,
Mhm.
later on, multiple smaller companies in between Mirati Therapeutics, Gossamer Bio, Trotana as VP,
Mhm.
and then at
Yep.
Takeda. But
Mhm.
essentially, my core discipline has been computational chemistry.
Yeah.
Um, and what is computational chemistry? Basically, it's an interdisciplinary field, I would say.
Okay.
So chemicals or compounds that we design or identify that can go and bind to proteins of target. So solving biological problems with chemistry, but using the principles of physics. The
Oh,
interactions of those compounds to the proteins of interest are guided by physics. So the laws of thermodynamics. So that is what
amazing.
comprises computational chemistry and its application to drug discovery. And there are
Okay.
many more nuances to it. How do we
Sure. Sure.
start from here where we identify those molecules or design those molecules, and
Mhm.
then we optimize them for their properties, their safety. So it's optimized for human consumption. And then, of
Okay.
course, it goes into clinic for testing. And so
Wow.
we are studying these molecules at a molecular level.
Yes, for
So
sure.
going to your
Yes.
next earlier
Yes.
question about explain structural chemistry and how you weave everything in.
Yeah.
So we start with the compounds or the ligands, but we also start with structures, which gives us a more three-dimensional perspective of
Okay.
how these ligands are actually interacting at a molecular level. So we
Okay.
start with the structures that are structure biology that comes up, that generates
Mm-hmm.
these models
Mm-hmm.
based on electron density or using cryo-EM.
Mm-hmm.
And then those give us an accurate picture, a snapshot, I would say,
Yep.
a
Okay.
snapshot of how these ligands may be interacting with the protein. They also give us information about the flexibility of these
Okay.
protein ligands. So use those. And how do we get to these structures is by generating the proteins first. We
Right.
produce the proteins. The proteins are then used to find the structure of the complex of the protein with the ligand,
Mm-hmm.
and
Mm-hmm.
get a molecular structure of here's a snapshot of how the protein binds with this particular ligand. Can we
Right.
now understand these interactions, and can we optimize these interactions? That's
Right.
the
And
field
we
starts.
okay.
And
Wow.
yeah, so
That
it's complicated, but it's very enjoyable, very interesting, and
sounds
yeah.
yeah. Well, I was going to say I heard all three of the like I heard three disciplines of science, biology, chemistry, and physics, all like kind of intertwined in the work that you do, and some of those maybe those levers are louder or higher at certain parts of your work than others, but it sounds like puzzle piecing together a lot of things, and maybe like I do with messages, you do with chemical compounds, like you're pulling them apart to see where they can be recreated to provide a different outcome inside of a chemical, uh, I don't know what the right word is, like solution for a drug, right? Like how, like
A
you're,
disease state.
yeah,
How do
okay,
we take care of a disease state by basically if a protein is overexpressed and leading to a disease state, then you want to identify compounds or molecules that can actually go and block that particular protein's action. So it
okay,
does not lead to that particular disease state. Those are
okay.
antagonists.
We're like
Or if
U-turn, make a U-turn, don't do that, right? That's great. That's
so,
great. Well, here, let me throw this at you because this is not in the pre-read that I shared about like what we might talk about. So you're gonna have to go with me on this. But just on Sunday or Monday of this week, I watched The Thinking Game. And I don't know if you've seen that, but it was one of the parts of the movie. It was about, AI, and I'm blanking on the name of this ridiculously famous AI scientist, but it was about protein folding at a
Mhm.
portion of the movie. What interaction, if any, is your work with protein folding? And like, help us understand that a little bit more while I look up the name of this guy.
Sure. So that's so proteins, as I said, are flexible. They're
Yes,
plastic.
right.
So that's what we also try to understand by structures. But
Mhm,
the other way to look at it, now that we have a significant number of structures that have been put out in the public domain, there
mhm.
are new algorithms, the AI-based or generative AI-based
Yep.
methodologies that have been developed now that we have the hardware capability to be able to fold these proteins now
Yeah.
successfully. This has been going on for many, many years, over 10
Mhm,
plus years. And
mhm,
many experiments failed initially, obviously, but now
mhm.
with
Sure.
more hardware capabilities and better
Okay.
algorithms, now we are able to actually fold these proteins.
Mhm,
When it
mhm.
comes to protein folding, we used to mimic earlier with homology models.
Okay.
We have known structures of related proteins. We do a
Okay.
sequence comparison and then come up with a model. This is
Mhm,
what we used to do earlier in computational chemistry. But
mhm.
now with generative AI um, and algorithms that can do it, we can now actually fold these proteins and even better, co-fold them with ligands that
Okay.
give us a perspective of, so structure is one way of doing it, getting the
Mhm,
actual data to be
mhm.
able to get a snapshot. But
Yeah.
now that these algorithms are now trained on those structures,
Wow.
you can now um, um, come up with a hypothesis or a model of how
Okay.
a ligand might bind to that particular
Okay.
protein, not just in its original site. I mean, there could be ligands that go to bind to some induced pockets, or they could go to some kind of orthosteric pocket. Some orthosteric pocket is easier because it's already a well-defined one. The
Yes.
challenge is more in, can the protein protein open up some newer pockets where these
Okay.
ligands can bind. And now
Right.
we can do it in a more rational,
Yes.
logical manner. Earlier, it used to be more serendipitous by high throughput screening. We found that it binds,
Hmm.
and we go from there. But now
Okay.
it's becoming more rational, more logical with the
Okay.
help of
And
these
it,
protein folding algorithms.
yeah. And it's accelerated your ability, the protein
Yeah,
folding and that's it helped you get further faster is what I
absolutely.
am understanding
It
from.
definitely has a very big impact
Yeah.
on
Right. Right.
progressing the discovery process, I would say.
Yes, for
Yeah.
sure. Okay. Hey, listeners, super quick station break question for you. Have you ordered your copy of my new best-selling book, Red Light, Green Light? Check it out. Grab your copy today at onpoint-communications.com/book. And now back to the show. Before we get to the red and green lights, I want to ask about a person maybe in your life who's really been influential for you. I know you came up doing, I think you were early interested in science from an early age and found you were really good at this. So, yeah, I'm always curious how people end up in the line of work or industry that they are an expert in. So, who do you kind of look at and say, that person really, or those couple of people helped me, and I owe a lot to their guidance or mentorship?
Mhm. Yeah. So, well, how do you get into that particular field is an entirely different
Right. That's another good question.
I'll start probably there. I was 15 when my grandma died, and that kind of
Okay.
motivated me to get into drug discovery even at that
Okay.
age. And since
Wow.
then, I've been working towards this goal. Parents, obviously, my family, my husband and son, they've
Mhm.
always been a very strong motivational force in
Mhm.
helping me to achieve whatever I could in my own goals and aspirations. Always been a rock solid support.
That's wonderful.
My PhD supervisor, B. Jairam, postdoc advisor, Don Bashford, and my first supervisor at Roche, Tara Mirzadigan, all of three were very
Okay.
instrumental in giving me the basis for computational chemistry and how to do it well.
Yeah, that's
They
great.
formed the basis, but then
Okay.
on to be a real drug hunter, Craig Coburn and Louisa Salter Sid at
Mhm.
Gossamer, who was the CSO, Craig,
Okay.
was my direct supervisor
Yep.
and VP of chemistry. And then at Takeda, Robert Goodnow, all of them helped me become a better drug hunter and a leader. So
That's
I
beautiful.
owe a huge
Yeah.
debt of gratitude to them
I
for.
love that. I love how you kind of peppered that with people from different parts of your whole life because, so often I just spoke at a high school a couple of weeks ago. I was asked to speak on like fortitude for these kids that are just getting started in life, right? And like looking at and what I find is like, we really can feel so kids, I think, feel a lot of pressure these days to know what they want to do, to have it all
Mhm.
figured out. And one of my messages was both, no, you don't need to have everything figured out. And to your recent point, like when you find good mentorship along the way, see it, notice it, and hang on to it
Yeah.
throughout your career journey so that you keep in touch. Because I've had, a list of people too, and hopefully all of our listeners can name a few that they're thinking of right now. But that matters, right? And you're probably paying it forward to others you've mentored, right? I would imagine that you've got
I
some.
feel blessed to have so many good people who've really been
Yeah.
a great support,
Yeah.
great mentors, and they're actually great friends too. I still
Yeah,
keep in
that's
touch with all
exactly
of them.
right. That's even more beautiful about it. That's
Yeah.
wonderful. Well, thank you for sharing that piece, Nidhi. Let's go to some of the red and green lights of scientific storytelling in your world. What does that look like when you're, who? Let's start with the basics here. When you think about some of your previous experiences, sharing the science, sharing the data, sharing the vision. You probably have done both non-science and science audiences. So just throw us a couple thoughts on some of those kind of red light things. Maybe those came up early in your
Mhm,
career, like it does for most of us, like, oh, that didn't work or that didn't land. Like,
mhm.
what's happening when scientists are trying to figure out the best way to tell the storyline? What are some of the roadblocks?
Yeah, quite a few. Ran into quite a few, but learned also by actually observing some good
Oh,
speakers.
I like that.
Yeah,
Okay. Yeah,
a
that
lot of
matters.
a lot of speakers that I have seen and observed. Okay, this is great.
Mhm.
But just boiling it down to maybe 2 or 3 points that
Yeah.
the potential red lights. One is obviously lack of preparation.
Okay. Yes,
So
for sure.
lack of preparation is one thing that leads to lack of confidence in how you present the matter,
Right.
nervousness that shows up. And I've realized over time that that nervousness gets mimicked by the audience.
Oh, how
So,
so? I love that.
it's kind of interesting. People see you as being nervous and they're bound to ask you more questions, more pointed questions,
Ooh,
trying to
goodness.
probe how much do you know about that particular
I love
topic.
that. That's so interesting. Nobody's ever said that. That's fantastic. And it makes total sense. I love
Yeah.
it.
So it's kind of interesting to note. So if you're prepared and you've thought ahead, then you're able to answer those questions in a
Yes.
more comes with experience too.
Of course.
So
Of
that
course.
that comes with experience. That comes with failing quite a few times to be able
Sure.
to realize, but also getting up to learn what did I
Yeah.
do not so well
Right.
and what can be done better. The other thing is that didn't work for me. Me meaning observing in
Mhm.
other presentations was a monotone in delivery.
Okay.
Tone matters
Yes.
a lot
Tone
when you
matters.
deliver the
Mhm.
message because the same message can be presented in a very aggressive, arrogant, and a
Right.
very soft manner. So
Mhm.
there's a very big difference in the impact that you get in the
Mhm.
way you deliver the message.
Yes.
So that's
Yes.
very important. But also if it is a monotonous presentation, people check out so
Oh,
fast.
it's so easy. Right. It's
Yeah,
really
and
hard to keep
the
attention.
best of presentations will go south
Mhm.
if it is a monotone. I mean, you
Yes.
may be giving out the best of the messages and the best innovation or discovery that you
Mhm.
maybe trying to bring about. People are not listening
Yeah.
because I like to say this, and the way I think it is, the audience there is saying it goes down to basic thinking. What's in it for me? The
Yes.
audience is thinking, why am I giving this person my time?
Right.
What am I getting in return? Am I listening to something interesting that would help me make decision about science and
Yes.
moving forward, some new discovery, some new knowledge? So
Yes.
if it is not delivered in a manner that can capture the audience or
Right.
keep the audience engaged, the
Yes.
message falls flat.
100%. I I feel drawn to make sure our audience understands the letters of my how-to model, which is rooted on the whole
Which
show.
is beautiful. I read
Yeah,
through your
you're
book.
hitting on it right now so hard because the how-to model, if you're new to our show, is an acronym, and it's really around what Nidhi is saying right now, which is that the audience has thoughts and expectations that they are asking of you from their seat, and they're never gonna tell you those things
Uh-huh.
unless you read my book. But, those things that they're asking you are H for hook me, get my attention. O is orient me, show me around, show me the landscape. What are we talking about? And why is it a problem? The W is win me. So win me over that you have like something legit to say here in this session that is a proposition. The word to in the how-to model is the T for tell me more. Tell me more about it. Now go to the data part. Now start convincing me. And then the final letter is O for offer me, like you said, like, what's in it for me? Ask me for
Yeah.
something. Ask me to do something. Ask me to
Yeah.
think something or believe something or vote for something or buy something or do something. So that's a tricky line to walk when you're the speaker and you want to share all your data.
That's I think the biggest problem with us as scientists is
Yeah.
because we worked so hard and we have a lot of data
Yeah.
to share. We want to give it all,
Right.
but that may not be the right way of doing it.
Right.
And that again comes with a lot of experience and a
Right.
lot of failures and also observing a lot of
Hmm.
people. My thinking is that imprecise presentation of the bottom line,
Yes.
the message is really a big red light that should
Yes.
be looked into by
Right.
every presenter.
Right.
And
Yes. I totally agree.
yeah,
Um,
and
Go ahead.
my thinking of green lights, if you want to convert them into
Sure.
green lights, just going to the last point itself is, or rather all of the points intermixed, is in terms of the monotone, the best is to break your presentation into smaller sections. I believe you covered that too, which I totally have
Yeah.
tested it out.
Mm-hmm.
Works better.
Yeah. Yes.
Breaking into smaller sections and basically making it a dialogue. Don't make it a monologue.
Yes. Yes. I love it.
Sometimes you may
that's
not
exactly what the book is about.
we are aligned on that. So, yeah, I mean, if it's not a dialogue, you're not engaging the audience. They're
Right.
just listening and they'll check out at some point. Then,
Yes.
as you say, they'll be on their phones.
Yes, they will.
Time is valuable. Everybody has and presenters should value that aspect because everybody's time is valuable. Why are they sitting in the audience listening to you if you cannot deliver something interesting that captures their interest?
Right.
They
Well,
will probably check out.
yes. And I want to share the word I keep hearing you say that I love is observing. Like I observed others and I learned what not to do or what to do. And it leads to one of the most simplistic ahas that anyone in the listening audience can do like right now, which is you can get better as a public speaker without ever opening your mouth.
Mhm.
Just watch people
Yeah.
and decide like, am I engaged with this speaker and what they're saying? Why or why not? Right? It's like those essay questions we had in high school. Why or why not? Are you checking out and disinterested? Why? What is the speaker literally doing, like behaviorally, that you should not do, like being monotone, going too deep in the weeds, staring at the screen behind them instead of looking at you, came unprepared? All those things, they really matter. And so even in the baby steps, we can get better just by watching other people. And
that makes a big difference. I mean,
yes.
in my life, it has made a big difference.
Yeah.
There is still a long way to go for me, I feel.
Oh, well, I think you're doing pretty well so far. Yeah. Like,
Hopefully,
yes.
but there is so much to learn. I mean, it's a whole wide ocean of knowledge, both scientific as well as in terms of learning from people. Every person that you meet can give you a good lesson that you can learn and hone on based on if you're receptive to it.
100%.
That's my thought.
Yes.
To that.
Yes, absolutely. Nidhi, when you think about where your industry is at this moment and kind of what's happening, I mean, obviously, I can't seem to have a guest come in without us talking a little bit about the impact of AI. And you were talking earlier about generative AI. What's happening where you sit that you can see that maybe would be fun, helpful, interesting for us to explore in your line of work?
Lots of things.
Okay. Bring it.
There are lots of wonderful things happening. I mean, so the
Yeah.
drug discovery process is divided into multiple phases, and what we are recognizing now is a lot of important innovations that are being made using generative AI, both in terms of, as you were talking about, folding and co-folding, which may be used for generating leads, but also in terms of optimizing our molecules so they can become better drugs, not just, I mean, the whole discovery process is pretty long. Overall, drug
It is long.
discovery takes about 10 to 15 years from a molecule that is a target that is being identified to a molecule being delivered to patients.
Right.
But the discovery process, we've already worked on expediting it to maybe 2 to 5 years.
Okay.
We can go through the discovery process
That's really
once we
good.
have a target. But with generative AI being applied at multiple stages, and perhaps agentic AI, which is now upcoming, we may be able to reduce or make the process more efficient and reduce the cycle time to maybe 1 to 3 years. That would be amazing. If 3 years it takes
Oh my
to
gosh, that would be incredible.
discovery, maybe in the 5, 7 years, we could basically 50% improvement,
I
50
love that.
or more improvement.
Oh,
So I think there's a lot of good methodologies that are coming up. Very
amazing.
excited about this field.
Yes.
My only concern would be that, or rather, I would encourage people to be more open and accepting these technologies, testing them out. They are going to fail. Some of them are going to fail.
Sure.
How we learn. And that's how we improve those methodologies to patients are waiting. That's my basic goal.
Oh, I like that.
Waiting for
Patients are
these
waiting.
medicines, and whatever we can do to expedite the process of discovery, I'm very excited about any methodologies that I can integrate in my work, and I would encourage every
Oh, I think you just
scientist to
gave
do
me the title for this episode, Patience and Waiting. I love it. I love that, Nidhi. Hey, listeners, super quick station break question for you. Have you ordered your copy of my new best-selling book, Red Light, Green Light? Check it out. Grab your copy today at onpoint-communications.com/book. And now back to the show. Well, I have two questions before we find out how people can reach you. And one of them is along the lines of, well, first of all, I just love like the crunching of the time frame. I just had another guest on kind of in an adjacent lane, but we did talk about clinical trials and the drug process and some of the things that he's driving in data management and
Mhm,
collection and analytics for companies. So, in the life sciences space, when you think about your whole career, like I think to myself, like, wow, it takes so long to get like a win in the wins column that's
mhm.
actually helping the patients. When you look back, Nidhi, like, what's one of your most proud things that you've been able to assist with developing or learning or bringing to the market, whether it's research or whatever else?
Yeah, if you want to know one, I would say there are multiple things, obviously, but
Sure.
I'm very happy about being able to contribute to the discovery of 6 clinical leads. 4 of them went to the clinic. 1 has been stopped recently, but 3 are currently in
Amazing.
clinic. The most advanced one was farmed out of that company to another company, but
Okay.
is 3 clinical trials
Incredible. That's
for prostate cancer. The other 2 that are currently ongoing are one for CLL and non-Hodgkin's lymphoma and lymphocytic leukemia, chronic lymphocytic
okay.
leukemia,
Okay.
and a third one going in for multiple sclerosis. I'm
Oh, wow.
very happy and proud of the whole team
Mm.
and
Oh,
my contribution towards
yeah.
the
Oh,
discovery
I
of.
have like chills. Yeah. I mean, to think that the work you're helping with really is for the patients are waiting. I love that. It's so true. And it's a reminder that sometimes we have to like lose some ground to gain some ground, right? Like you have to try this stuff, like you're saying, and there's ample reason to move forward with new ways of doing things if it can really bring us a leap and
Yeah,
cut in half the time that we can get a drug to market or do the research. Like obviously have to be careful of all those things, but it's still it's a very exciting place to be right now with all that's coming through
oh,
generative AI and other discoveries that are helping you do your job better.
right.
So that's fantastic. My other question has to do with the reality that because we work so much with these PhDs and researchers who have spent their lives, you have spent your life up until you get to your first like quote unquote real job, right? In the annals of academia and
Mhm,
researching and writing papers. And I was on a call with a leader that I'm engaged with in a large team. And he said, Cindy, we are, and he's now in a sales position for this company. Okay. And it's like, we have spent our whole careers so far, for the most part, explaining and
mhm.
proving and
Justifying.
showing. And now you and my company are asking me to listen deeply to my customer and to shut my mouth and not blather on not blather, but, like, not say all the things I know, and he said he was being very open about the knowledge that that's a switch he needs to make, right? But how hard it is more my point, like,
Mhm,
yeah, I was like, "That is brilliant, actually," because you're right. Researchers are like that you cut your teeth on proving, and now when you arrive in an organization that, you're trying to do more of selling and talking to customers, you talk less and listen more. Have you experienced this shift in any way? Yours
yeah, I have. As you grow more into the leadership, um, and you're talking not just to your peers or to your subordinates, or to your mentees.
Mhm,
It is also conversations with the CSO or the president of the
right.
company.
Mhm.
Um, but when you do that, yes, you have your own deck of things and thoughts
Mhm,
of what you want to share with them.
mhm, mhm.
But point is also about knowing or understanding what is it that they are looking for.
Yes.
So
Yes. Think like
I've read
vaping.
about it earlier. I've observed that earlier with people, but I've also read about presentations and messaging.
Yeah.
Is that you need, and you say it very nicely in your book too, flip
Mhm.
around and sit in that audience's shoes.
Right.
And
Right.
it's more so for people who are more senior than you. They have
Mhm.
very less amount of time, and they want
Yes.
to get maximum amount of information that can help the business, that
Yes.
can help them moving forward, or
Yes.
if they're presenting something, the information that they should be boiling down to.
Mhm. Yes.
So
Oh, I love it.
again, it comes down to in your mind, and that's what I think is having not just the depth of that knowledge, but
Mhm.
also a clear bottom line.
Right. Yes. Help me
If,
help you.
yeah,
Right.
let me help you. So if you have the bottom lines clear in your mind, if there is no time, let's say, give me 2 minutes. What do you want to say?
Yeah.
Here are the key things that I want to communicate.
I love it.
And if they need more information, they will be more open to asking that, and then we can fill in the details for
Right.
those.
Right.
It's not easy to do that. Again, I said you have to work on that. You have to
Right.
fail a few times, but still be able to be open to gain that
Mhm. Yep.
information, or what is it that they are looking for? What
Well,
is
and
it that they can
yeah.
share with them that would help them move forward and
Exactly.
help science
Exactly.
move forward?
And to harken back to one of your earlier points, it's about the delivery of your 2-minute type message to the CSO or the CEO, where you can bring your gravitas and your confidence in the 2 minutes you have brought and deliver it in a way that just like lights up the recipient to say like, I know if I have another question, this person has the answer. And it's
Hmm.
just like this thing that travels in midair, right? Where you
It's
don't need to
trust. It's
say it.
building trust.
Right? Yes, trust. Exactly. Well said.
Yeah.
Oh my gosh, Nidhi, this was even just as wonderful as I knew this conversation would be. And unfortunately, we're already out of time, which is not
That
fair.
went fast.
That was so fast. But I really have enjoyed this conversation and going into some of the underground areas of computational science, protein science, and the incredible work you're doing kind of under the hood, I would say, from what most of us out here in regular life land like really observe. So
It's
thank you.
regular. It's
Yeah.
absolutely regular, and
Yeah.
it's fascinating. It's fun and very
Yes.
exciting to be able to help. But I
Oh,
thoroughly enjoyed the conversation.
yes.
Thank you for bringing me
You
on,
are
and
so
it's
welcome.
been
Do
so
you
great.
have any like quick one thought of advice for a new scientist coming into the field, like out of grad school? What do you say to that individual
Mm.
who's about to start a journey like yours?
Stay excited about
Yeah.
science and keep the goal clear. Work hard,
Yeah.
and learn to have that clarity of presentation by
Mm.
knowing yourself, your
Mhm.
subject matter, and your audience.
I have nothing else to say, my friends. Fantastic. Diddy, thank you. This was just so much fun. I really appreciate you being here with us today. You're going to bring great value to the listeners here. And maybe we'll bring you back again for another round two.
Amazing.
That would
Would
be
love to
amazing.
do that
Yeah.
too. Thank
Yeah.
you for
You're
inviting me
so
again.
welcome. Thank
Yeah.
you. Thank you. Thanks, everybody, for listening. Thank you for watching. Make sure that you hit like and share and send this to a scientist near you or someone who would just be interested to learn more about what we talked about here on Red Light, Green Light. I will be back next week with another fantastic leader to share their stories with you on their journey to becoming a top 5% executive communicator. In the meantime, get out there, find yourselves an audience of stakeholders, move them from the red light of risk to the green light of go on your message and nail it. Thanks, everybody. Bye bye.